Hello class, good afternoon. Today we will be learning about C. elegans. They are more commonly known as the roundworm and they belong to the phylum Nematoda. Yes, Rohit.
Worms? What's so great about worms? I don't want to learn about worms!
Wow, that's a really great question, Rohit. Let this voice answer that for you.
C. elegans are roundworms of the phylum Nematoda. This phylum contains both parasitic and free-living worms that have evolved to inhabit nearly all environments on Earth. C. elegans are hermaphrodites, non-parasitic, and are found in temperate soil, where they feed on the bacteria in the soil. They are microscopic and were the first multicellular organism to have its entire genome sequenced, where 40% of the genes have human matches, and are a very commonly used model organism in neuro and developmental biology.
That might be true, but I don't know, they still sound boring to me.
On the contrary, Rohit, C. elegans are very unique and are often used to do many different types of research.
The idea of using C. elegans as a model organism rose over 30 years ago. In 1998, the entire genome sequence of each cell of the organism was found, which was led by John Sulston. Many factors contribute to why C. elegans can be utilized as a model organism so effectively, including their short life cycle, ability to self-fertilize, transparency, and rapid reproduction. The life cycle of C. elegans spans about 3 days from zygote to sexually mature adult. This makes them easy to quickly prepare and allows many induced mutations to be assessed in a short time period. The ability of C. elegans to self-fertilize makes them an effective model organism because an entire population of C. elegans can be obtained from one single plated hermaphrodite. Each C. elegans is capable of producing up to 300 offspring at a time. The ability of the hermaphroditic C. elegans to self-fertilize makes them particularly useful in maintaining homozygous mutations due to the fact that crosses are not necessary to reproduce.
Wait, the life cycle is only 3 days? How's that even possible?
Well, Rohit, the process is actually quite interesting.
C. elegans' life cycle consists of an egg, 4 larval stages, and an adult stage, and the entire cycle occurs in about 3 days. The worm molts after each larval stage to allow for more growth and development. The adult is a hermaphrodite, so it has both sperm and eggs, and is the largest of all the stages. The adults release the embryos through their vulva, which is the egg-laying structure located near the middle of the body. The embryo is a small, circular, or oval structure. Embryonic development occurs for about 14 hours until the first larval stage. Each larval stage has the shape of the adult but gradually increases in size as the stages progress. The L1 stage is an embryo that has just hatched and is the smallest of the larval stages, and is in the stage for about 12 hours. The L2 stage is a little larger than L1 and lasts for about 7 hours. The L3 stage, which lasts about 8 hours, then molts into the final larval stage called the L4 stage. The L4 stage has a characteristic crescent-shaped clear patch, which is the area where the vulva will form. The L4 larva molts one more time to become the adult, where embryos can be seen inside through the use of a microscope. The Dauer pathway, which is a dormant stage, is an alternative pathway for development that occurs at the end of L2. The worm takes this pathway when under stressful environmental conditions. The Dauer stage is non-aging and is characterized by reduced movement and arrested feeding.
One unique trait about C. elegans is that there's no female sex.
No chicks? How does that work?
Hermaphroditic C. elegans are able to mate with males or self-fertilize. The presence of males is rare, and usually the male population is only 0.1 to 0.2% of the overall population. This is due to the fact that males arise from rare non-disjunction of the X chromosome in crossovers. The majority of self-fertilization offspring will become hermaphroditic, but crosses with males will yield more male offspring since half of the sperm are missing an X chromosome. The hermaphrodites and males have different characteristics. Hermaphrodites contain 959 somatic cells, while males have 1,031 somatic cells. Hermaphrodites produce sperm early in their life cycle and then store embryos in their uterus at a later L4 stage. The L4 stage is the first life cycle stage at which hermaphrodites and males can be differentiated from one another.
Some very interesting stuff. Now, how would you like to learn about fertilization?
Hell yeah.
Life begins for the C. elegans when sperm enters through the spermatheca and fertilizes the oocytes. In the adult hermaphrodite, the oviduct, spermatheca, and uterus are all next to each other. The first contraction allows for the matured oocyte to leave the oviduct and enter the spermatheca, where the sperm is held. A second contraction squeezes the fertilized egg into the uterus. The excess sperm are able to retreat back to the spermatheca. This is fertilization for hermaphrodites. Fertilization is slightly different for males. The males deposit their sperm near the vulva of the hermaphrodite. These sperm then move to the spermatheca, where they are preferred for fertilization of the matured oocyte over the hermaphrodite sperm.
And after fertilization, an event called cleavage occurs.
Oh, cleavage. I've heard of that one before.
No, Rohit. Not that type of cleavage. This kind is better.
Cleavage in C. elegans is a highly choreographed process where they undergo asymmetric, rotational, holoblastic cleavage. With each early cleavage, the zygote divides into a founder cell, which forms the blastomere, and a stem cell, which forms the germline. During the first cleavage, the cell divides into the P1 stem cell and the AB founder cell. As cleavage continues, the daughter cells are named relative to their position. The AB founder cell divides into ABA, which is the anterior daughter cell, and ABP, which is the posterior daughter cell. Each of these then divide into the right and left cells. The AB founder cell forms the pharynx, hypodermis, and neurons. P1 divides into P2 and EMS. EMS then divides to form E and MS. P2 divides into P3 and C. E forms the intestines, while MS forms muscle, pharynx, and gonad tissue. C will form the muscle, neurons, and hypodermis tissue. Finally, P3 divides into P4 and D. P4 will form the germline, while D forms muscle tissue. The beginning of gastrulation is marked by the inward movement of the E founder cell from the surface. This inward movement is also known as ingression and requires the presence of certain proteins. After the inward movement, the empty space left at the surface is filled with the neighboring cells, which are MS and P4 cells. Gastrulation advances as ingression continues.
So to end today's lesson, I would like to talk about mutations.
Mutations? Like the X-Men?
Not quite, but pretty close.
Mutations are commonly used for developmental research, and as a result, many mutant strains have been made. Some common mutations include the gene families of dumpy, blistered, and uncoordinated. These gene families consist of many genes. Dumpy is characterized by being shorter, fatter, and slower than the wild type. Blistered is characterized by fluid-filled pouches covering the surface of the body structure. These blisters greatly inhibit movement and can be lethal if severe enough. Uncoordinated is characterized by abnormal movements. There are multiple ways the movement can be mutated so that the worm does not move in a regular, forward, snake-like pattern.
Wow, I never thought I'd say this, but worms are pretty cool.
I'm glad I've changed your mind, Rohit. Now go out there and share your knowledge with the world.